Hello, everyone. My name is David Jenkins. I'm a medical microbiology consultant in Leicester, England, and I'm one of the co-chairs of today's webinar produced by the International Society of Antimicrobial Chemotherapy. It's a webinar on bacteriophages, and we've got two expert speakers talking about some really interesting aspects of bacteriophages and their potential use in clinical medicine. And this is a really exciting part of new therapeutics of infectious diseases. So I'm really happy to be able to be part of this webinar. Before we get started, I want to introduce my co-chair, Michael David. Michael, over to you.
Hello, thank you, David. My name is Michael David. I'm an associate professor of epidemiology and infectious diseases at the University of Pennsylvania in Philadelphia, Pennsylvania in the USA. It's my pleasure to be here and co-chair this webinar, which I think you'll enjoy very much. Before we start, I'd like just to say a little bit about the International Society of Antimicrobial Chemotherapy. It was founded in 1961. It's a federation of more than 100 affiliated member societies. The goal of ISAC is to increase the knowledge of antimicrobial chemotherapy and to combat antibiotic resistance around the world. ISAC promotes and hosts the International Congress of Antimicrobial Chemotherapy, the ICC, and the next meeting of the ICC will be in Manila from the 10th to the 12th of November, 2026. So ISAC sponsors two international journals, both of which have very interesting content and are leading journals in the field. They are the Journal of Antimicrobial Agents and the Journal of Global Antimicrobial Resistance. So ISAC sponsors a great number of working groups which drive forward the work of ISAC in very specific areas of interest to infectious diseases and antimicrobial therapy, and they're shown here. ISAC also sponsors the ISAC Academy, which is an online educational resource which hosts both ISAC and stakeholder educational material on key infection concepts, and the website is at the bottom of this slide. We invite you to explore those resources. Also, ISAC sponsors the Antibiotic Guide, the ABX Guide, which is an open access online antimicrobial treatment guideline for global applications. It's intended to provide guidance on antimicrobial treatment of common infectious diseases. It has an easy-to-use modular format, and it can be adapted to various settings. The main aim is to address gaps causing inappropriate antibiotic use, particularly in low-resource settings. So it's a unique resource, and again, we encourage you to explore that. The website is shown at the bottom of this slide. Finally, ISAC has merged with the Alliance for the Prudent Use of Antibiotics, or APUA, in 2019 after the retirement of the founder of APUA, Professor Stuart Levy. And so we encourage you to follow ISAC on social media to keep up to date on new activities, including those that I just mentioned. And now I'd like to pass it over to David to introduce our first speaker for the webinar.
Great, thanks very much, Michael. So it's my great pleasure to introduce the first speaker, Dr. Aycan Gundogdu, who is from the Erciyes University School of Medicine in Turkey. She is at the Genome and Stem Cell Center. Dr. Gundogdu has a PhD in medical microbiology from the University of the Sunshine Coast in Queensland, Australia, and where she served as a postdoc researcher as well. She's been actively involved in metagenomics, microbiome, and of importance for this presentation, bacteriophage projects. And she's worked at a number of fantastic institutions around the world. Apart from Queensland, she's also worked at the Commonwealth Scientific and Industrial Research Organization, the Eliava Institute in Tbilisi in Georgia, which is, everybody who knows anything about bacteriophages will know it's the epicenter of bacteriophage work globally. The Federal Institute for Risk Assessment in Berlin. She returned to Turkey in 2015 and played a key role in founding the metagenomics division at Erciyes University. And she's managed several national and international projects. And as I mentioned already, significant work in bacteriophage research at Eliava, and co-founder of Nbiosis Biotechnology, which is a spin-off enterprise from her lab set up in 2019. And she's currently the Chief Scientific Officer there. She takes part in a number of national and international panels, including the Turkish Scientific and Technical Research Council. And in 2021, she was appointed as a member of the General Executive Board of TÜBİTAK. So her team's research primarily is into the discovery of biomarkers from disease microbiomes, manipulating microbiomes, studying viromes, constructing bacteriophage libraries, and implementing phage therapy. So these are all at the cutting edge of antimicrobial development these days. It's amazing. So Dr. Gundogdu is going to be talking, sorry, I just missed that bit, is going to be talking about phage therapy, how to build a bridge between researchers and clinicians. Aycan, over to you.
Thank you very much. I would like to thank to ISAC again for inviting me. It's a great pleasure. Today I am going to give a talk on phage therapy. I mean, we can see that there is an increasing interest in phage therapy, and parallel to this interest, research on phage is also growing. I mean, researchers in the field are putting into a great effort for the preclinical aspects of phage therapy. But despite this, we cannot see the reflection of that effort in clinical side. So in this talk, I will try to discuss, what is kind of lagging this translation. So let's start. I'm going to give just fundamentals about the phage. I am sure everyone knows about it, but I just would like to give some basic information. All we know, phages or bacteriophages are viruses that specifically infect bacteria. They usually target specific bacteria species or even strains. And the phages are the most common biological entities on Earth, and they, found everywhere, even in our body. I mean, phages are part of human microbiome, which means that we are constantly actually exposed to them. We are constantly exposed to phages. And the phage therapy, which involves using phages to treat bacterial infections, but I'm talking about the lytic phages, not the lysogenic one. And then it dates back to 1919 and quickly gained popularity after that. So in its early days, like second quarter of 20th century, phage production and clinical use were widespread globally. And this is the Eliava Institute, one of the prime example, as a phage center. I think everyone who works on phage therapy, or at least who is interested in phage therapy, know this place. But just in case, let me give a brief overview. It's established in 1923. It has been producing phages nonstop ever since, and research, phage production, as well as phage-based treatment continues there today. And here are some other centers and companies that produced and sold phages during the early years of phage therapy. And I have to say that not only institutes or hospitals, but also many pharmacies produce and sold their own phages to patients. So in these early days, for a while, phage therapy was successfully applied. Unfortunately, things didn't always go smoothly, and eventually a kind of phage therapy winter arrived. I mean, there were multiple reasons for this, but I don't just have time to detail them all. But today, when we look at the back, what was the main problem? I can say that the first one, the early phage biology and the production techniques were poorly understood, and this led to issues like difficulty in distinguishing between lytic and lysogenic cycle. Attempts to use phages against viral infections, which are definitely not effective. And also, early phage preparations were often treated with harsh chemicals like mercury, or treated with heat, which reduced their effectiveness, of course. And also low purity and contamination issues like endotoxins further limited success and lead to inconsistent results. So, and also in addition to these challenges, the early stage of phage therapy lacked well-designed clinical trials to establish its efficacy or prove its effectiveness. And antibiotics, another problem, and because antibiotics had a major contribution to the like phage therapy winter. I also have to mention the reports from American Medical Association because of the controversies surrounding early phage therapy. The American Medical Association requested Eaton and Bain Jensen to analyze all available phage therapy data and prepare a report, and then they did. And this report was published in JAMA Journal. According to this report, there was no evidence providing that bacteriophages were viruses. And also the antibacterial effect observed was likely due to an inert substance. But as successful results continued to emerge, the American Medical Association this time asked like different scientists to evaluate phage therapy studies, and this analysis was published in like JAMA again. According to their analysis, phage therapy had no significant effect compared to available drugs. So I can say that these two meta-analyses like challenged early phage therapy, criticizing the lack of rigorous evidence. And finally, the Second World War put the, I think, finishing touch, and the geopolitical divide of the Cold War widened the gap. So the phage therapy continued in the Eastern Bloc, but was mostly abandoned in the West, where it was linked to Stalin and replaced by like widespread antibiotic use. So eventually phage therapy itself was nearly forgotten worldwide. But I have to say that, or I emphasize that I am talking about phage therapy, not phage, because we have been using phages for other biological purposes. So, and today in the search for solution against antibiotic resistance, luckily phage therapy has been rediscovered, and the researchers from the former Eastern Bloc played an important role in bringing phage therapy to the West. So this reintroduction started new interest in phage therapy. As a result, we started to see some sporadic cases of phage treatment began to publish in West, and one of the famous cases is the von Peterson cases. Probably most of you know this case. He was critically ill with multi-drug resistant Acinetobacter baumannii infection, and phage treatment administered IV in the US, and this led to his rapid recovery. So this case attracted significant attention, and phage research increased even further after this case. We can say that clinical studies have increased over the past 7 or 8 years, but still limited. I mean, despite the increase in phage research and recent advances, clinical integration remains limited. I would like to share one of my how can I say observation. This is Eliva. This is Tbilisi, actually, not Eliva. Sorry. I mean the clinical integration limitation. I mean, I can understand this situation in the West. But I expect Georgia, where the Eliva Institute has been active for 100 years, to be an exception. I mean, but however, I recently realized that I was wrong. I mean, what I mean that, okay, I said this is Tbilisi. I was there last September as a part of project. Here is the Eliva Institute I mentioned earlier. As I said, it has been dedicated to phage therapy for over a century, with patients coming from all over the world. And this is a urology center in Tbilisi, where they recently published results from clinical trials on phage therapy. During my visit, I met Dr. Alexander, who is the physician responsible for these two studies they just published. It's a very big clinical trial, actually. And actually, yeah, what struck me most during our conversation was when Dr. Alexander shared that he initially faced considerable resistance. I mean, in the first year of his study, no clinicians was willing to refer their patients to this study. And he noted that there were around like 40 urologists in this clinic, and none of them wanted to refer patients to his study. I was very surprised because I had assumed that the doctors in Clinic Tbilisi easily prefer phage therapy over antibiotics. But it turns out that wasn't the case. But why? I mean, if they face clinic challenges, bridging lab and clinical practice in Georgia, how we will bridge, this gap in West? I mean, where phage experience is far more limited. This was the main question actually in my mind. So I believe the answer lies in understanding clinicians' perspective on phage therapy. So I have grouped clinicians into 3 categories, but you might think more or there might be others. The first group has no knowledge about the phage therapy. Despite growing awarenesses, I still met clinicians who haven't heard of it. And the second group is aware about the phage therapy, but skeptical due to past failures or concern about resistance, bacterial resistance against phage. And the third group is aware, interested, and willing to use phages, but faces significant real-world barriers. So, I mean, how we can address the concerns of these groups, or how we can make a bridge between clinic and the research. The first group, I believe, the raising awarenesses, I believe that if we can address the concerns of the other 2 groups, awarenesses in this group will naturally increase as well. So I think we have to focus solving other, 2 groups' problems. The major barrier to adopting phage therapy, I think, is trust. Many clinicians who are aware of phage therapy are often skeptical. As I said earlier, even in Georgia, same problem exists. But Dr. Alexander mentioned that once initial results showed phage therapy has to be as effective as antibiotics, the entire situation changed. And clinicians began to approaching him and eager to include their patients in the study. Sorry. Okay. So I believe that restoring confidence in phage therapy requires rigorous multidisciplinary clinical trials and collaboration between microbiologists, pharmacology, and clinical trials would lead to reliable, reproducible, and clinically relevant results. But at that point, I have to say that let's not allow history to repeat itself. What I mean that isolation of phages seems very easy and at first glance, of course. I'm collecting environmental samples at targeted bacterium, incubated overnight, then centrifuge and filter, drop the filtered samples onto petri dishes with bacteria, and if you see clear plaques the next day, okay, there is a phage. And this is one of my petri dishes. So I tested 4 different water samples against MDR Klebsiella, including colistin-resistant strains, and found 2 of the water samples contain targeted phages. I mean, okay, this might seem straightforward, and in some ways it is, but it is also a kind of double-edged sword. I mean that this simplicity was one of the main reason why phage therapy lost its popularity and faced winter. And actually, I am a little bit worried similar issue may arise again. I mean, as all we know, essential steps like purification, concentrations, endotoxin removal, stability testing, are critical to ensuring phage cocktails are safe and effectively. And I know that today, everyone is aware of these steps, but I would like to emphasize that Each of these steps is kind of like an art. I mean, each step requires skills beyond simply following the literature. And we see that even like big projects sometimes overlook basic details. So I would like to underscore that not every phage is suitable for therapy. So, and one of the issue of bacterial resistance, a common concern is, okay, phage is not nice, but resistance develops quickly. This is like true, but I think this is only partly true. What I mean that phages have a unique advantages over antibiotics. They co-evolve with bacteria. And this adaptability means we can develop new or modified phages to target resistance strains. This makes phage therapy a dynamic alternative to static antibiotic treatments. Even like when resistance occurs, it often wakens the bacteria, making them more fragile to the immune system and antibiotics. Additionally, like phage cocktails, like combination of different phages targeting the same bacterium, make it much harder for bacteria to develop resistance. And also, when we look at the real life experience, phages have been using more than like million people, and no significant resistant trends were observed. So in reality, like bacterial resistance to phages is manageable, especially compared to antibiotic resistance. But we should be in a time where we no longer talk about these simple details as a problem, because I believe that we don't have the luxury of ignoring phages, but we are not outside of the science that we should exaggerate them. I mean, we just need to recognize their real potential. For the last group, the eager one, I think there are two real-world barriers, and of course, the first one is regulation. In many countries, there is no clear framework for phage therapy. Of course, some countries are a step ahead. However, in many countries, there has been little progress in terms of regulation. I mean, should phages be regulated as drug or treated different like biotherapeutics? It hasn't been decided yet. So we definitely need clear regulatory guidelines. But what makes regulation more difficult, I think, the existence of two different approaches. I mean, there are two approaches to phage therapy. The first one, ready-to-use products, and the other one, like customized phage treatment for individual. And each require different regulation, so a single framework is challenging. I mean, a one-size-fit-all framework, a little bit challenging. So I think a dual-path regulatory system, like with flexible but clear regulation for personalized treatment and rigorous but straightforward protocol, the ready-to-use one could be the solution. And also, I have to mention the lack of standardized protocol here. Unfortunately, there are no established standards in the phage therapy. For example, for phage stability testing, some researchers use Phage assay, others use time kill assay. So for successful phage therapy, and of course regulatory approval, we need clear, universally accepted protocols for phage isolation, production, and clinical application. And also establishing international standards for phage therapy is essential and should be developed through collaborations led by global consortia. I absolutely believe in that. And the other problem, the last one, even if regulation is no longer a challenge, how will clinicians access phages? I mean, there is still limited phage library and accessibility. This is, I think, the main problem. Currently, a few universities and research centers have their own phage libraries and production capabilities. And I don't believe this model is sustainable or efficient for broader demand. Because as all we know, unlike antibiotics, phages have a narrow spectrum of activity, and they are live biotherapeutics. So which means library diversity, production, and logistic presents unique challenges, different from like antibiotics. So I have to say that the limited accessibility to comprehensive phage libraries remain a like pressing issue. To address this challenge, I think centralized phage production facilities integrated into the like national healthcare system could provide a forward-thinking solution. I mean, these facilities would act as like kind of central phage banks, and they would be regularly updated and accessible to clinicians across the region. And high standards and sustainable practice would ensure these libraries remains reliable, scalable, and adaptable to emerging bacterial threats. So, in conclusion, phage therapy holds immense promise in the fight against antibiotic resistance. I think everyone accepts this reality, and its implementation faces hurdles like trust, standardization, regulation, and availability. But I think each challenge has a viable solution, like starting from building trust and like centralized production. And I believe that with systematic coordinated action, we can build a bridge and position phage therapy as a reliable and accessible approach against antibiotic-resistant infection. I have to say that there is like a long to go, but let's not allow history to repeat itself. Instead, work together to ensure the phage therapy research, phage therapy reach its full potential as a life-saving tool in modern medicine. And thank you for listening.
Thanks very much indeed for a really fascinating presentation. Thank you. Over to you, Michael.
Thank you, David. Aycan, thank you very much again for your presentation. I agree, it was wonderful. Our second speaker today will be Professor Krystyna Dąbrowska. She is a full professor at the Institute of Immunology, Experimental Therapy at the Polish Academy of Sciences. She's also a professor at the Wrocław University of Science and Technology. Dr. Dąbrowska received her PhD from the Institute of Immunology and Experimental Therapy of the Polish Academy of Sciences, and she also holds a postgraduate degree in project management and EU law from the Wrocław University of Economics, Department of Management, Informatics, and Finances. Her research interests are really diverse, having to do with phage therapy and phage science. She studied immune responses to phage treatment, specific and innate immune responses to phages, and phage pharmacokinetics and the effects of phage on mammalian systems and cells. She's also studied molecular factors that impact the effects of phage. She's very well published and on all of these subjects. And so today I welcome Dr. Dąbrowska, who will speak. The title of her talk is Phage Pharmacokinetics, Why Does It Matter? So over to you, Dr. Dąbrowska.
Okay, so I would like to say hello to everybody. It's a great pleasure to have this opportunity to talk about phages that are fascinating objects, and I'm fascinated with phages I've been fascinated for around 25 years, so it's a huge time. Basically, I come from the institute that has a nice history of phage treatment, even within the times where phage therapy was, as we call it, forgotten. And this is Hirschfeld Institute of Immunology and Experimental Therapy in Wrocław, Poland. And the other institution, it's a modern one, medical faculty, University of Science and Technology. But my phage experience comes from the first one. So I would like just show where I'm broadcasting from. So this is right here, Southwest Poland. And I invite everyone to go sightseeing in this place. Let's talk today about the topic that is very important in phage therapy. Because when we think about phage therapy, we should try to find the way how to make it most efficient. But it's not the only way, and it's not the only topic that we could investigate thinking about phage, because these guys that you can see right now in their action, they are probably our future drugs that could combat very difficult bacterial infections. But very importantly, phages are everywhere in nature. So we are constantly exposed to phage in food and in drinking water, and they just live inside human and animal bodies as a part of our natural microbiome. So it's a regular thing that we are exposed for natural phages, and we would like to increase it a little bit by exposing specific patients to therapeutic phages. So it's a very significant thing that we should think about combining these two elements. Okay, so what happens when a human body meets a phage? So primary reaction of human body to foreign objects is defense, because one never knows, maybe it's a pathogenic object. And what protects us, these are anatomical barriers. This is then like the second line, innate immunity, which is not specific. And at the end of the day, we have adaptive immunity, which is specific, but of course it must be trained before we can get this specific immunity response to any objects. And all these three lines of defense apply to phages as well. So we have to overcome, we have to take into account, and when we plan our therapeutic schedules, it's good to know that we will have to face somehow and to think how to overcome this defense aspect of our body. So anatomical barriers, they are probably very well known, and this is everything what applies to bacteria and to pathogens. So it's no surprise that we have just typical anatomical barriers, like for instance skin, but also pH or enzymes or other elements that protect us from entering our body by many microbes. So this brings us to the question, what is the most efficient administration route? Because as we see, depending on the route of administration, we may get different chances for delivering phage to our body. And what I would like to present here, these are results of systematic review that was completed quite a few years ago, but already on a big number of reports that came from phage therapy in animals, or just phage experiments in animals. And taking all these experiments together, we could observe that one thing that is very easy, this is gut transfer. So when we give phage orally, it's almost 100% chance that the phage will travel to the other side and it will be active. Even if we'll lose some phage due to the gastric acidity, it's not a disaster for the phage usually. So we can improve it a little bit, but still, even if we don't do anything, in many cases we can expect that we get quite a nice gut transit of a phage. However, when we consider absorption, which means how much phage will be absorbed and distributed around the body when we give the phage orally, it's not that good, because oral application is the least efficient way of application phage for distributing it around the body. So what is most efficient? What is more efficient? This is topical application, which might be a little bit unexpected, but it's around 50% of efficiency. Then when we inhale phage, of course, the phage that has been inhaled to our lungs, to our airways, it can work just right there, but it can also be absorbed and distributed around the body. And this is more or less 2/3 of cases that confirmed efficient transfer of phage by this way. And of course, injections anytime. Usually they are super efficient and we just get what we inject. So the phage is immediately distributed around the body. And usually this is the best way when we want to make sure that we can deliver the phage to the system. So one thing that is limiting our availability of phage and how the phage circulates around the body, this is how we give this phage to our patient. Once the phage enters the body, it will encounter many elements of our immune system, and it will be recognized by the immune system elements. So the first line that is always ready, this is innate immune system. And for instance, it's represented by phagocytes or by the complement system. Later, when we challenge long enough and with enough dose, then we may teach our immune system to recognize the phage specifically. And this is the adaptive immune system that is represented by antibodies, T and B cells, T and B cells that can recognize the phage specifically and that produce these specific elements like antibodies. So the first one can be linked to inflammation. So in this part of the response, the inflammation is the major tool for the body to get rid of microbes. And here we have, and in the adaptive immune system, we have specific recognition, which is linked to a very important part, which is immunological memory. So what is responsible for phage clearance, taking this innate immunity part? So this is the liver and spleen that have been many times demonstrated to filter the phage, to harvest it super efficiently. And when we consider if they can or cannot inactivate phage, then we see that, for instance, splenocytes may play a very significant role in the phage removal. What you can see here is the amount of phage that survived after incubation with splenocytes. And in blue, you may see just normal, non-activated splenocytes. And red and green, these are two versions of activated splenocytes. The red one, these are splenocytes that have been isolated from mice with systematic inflammatory response, which means like high level of activation for immune cells. And the other one, the green one, this is PMA activated splenocytes because we can activate them chemically as well. So as you can see, we have a difference and we have direct effect of the immunological state of the splenocytes and how much activated they are. The more activated they are, the more efficiently they remove phages from their environment. And here we have also active ones, but not that much. So let's think once the phage is engulfed by innate immune cells, what may happen then? So the major pathway for phage engulfment, this is macropinocytosis, because the phage is pretty big. So this is a pathway that is available for that size objects. And what is also very important, it does not require specific recognition or even not specific, but pathogen-associated molecular patterns are not required to engulf an object. So anything can be uptaken this way. However, we can also observe that phages are engulfed by phagocytosis because it also allows the cells to engulf pretty large objects. However, here we need specific recognition, we need a receptor. And when we don't have receptors that are dedicated to phage, we should take into account that it can be mediated by Fc receptors that can recognize antibodies, which means that once we have antibodies that circulate and recognize phage in our body, then this pathway of phage engulfment will also be available. There are rather rare observations that clathrin-mediated endocytosis can also apply to phage. However, here we have just more strict restriction of the size. And for this reason, we can it can apply only to small phages. Same about cavallar endocytosis. Pinocytosis has never been demonstrated and still not recognized other ones. What happens when phage is inside the cell? Then there are two possibilities. One is that the phage in the vesicle will be directed to lysosome and to degradation. However, they can be just transcytosed to the other side of the cell. And this is the main mechanism that allows phage to enter our body via our mucosal surfaces. So these are activities of gut cells, for instance, or cells that are actively transporting phage in the airways. Very shortly, why not excretion? This is what we can learn from quantum dot models, because on phage this is super difficult. They are so diversified that the best way of demonstrating that these are quantum dot models. And here what you may see, this is a quantum dot model completed in mice that present a relation of how big the object is and does it tend to be removed by urine, or is it rather deposited in the tissues and engulfed by the cells that will then degrade it? And as you see, around here, this is between 5-6 nanometers, which is still way less than phage. We have this switch that urine excretion is less and less important for an object removal. And same here in the animals, you may see that smaller objects, they are rather transferred and excreted to the urine. However, they are deposited in the tissues, liver, spleen, lungs, when these are bigger objects, still way smaller than the phage. So, of course, the phages can penetrate to urine, but it's not the major pathway for phage removal from the body. And what happens when this phage circulates in the body long enough? The phage induce phage-specific antibodies. What you may see here, these are the levels of IgM and IgG antibodies in animals that were challenged with two different phages. Here, the upper panel, F8 phage. Here, the lower panel, A3R phage. Challenges were marked with arrows, And here at the x-axis, we see days after the challenges. So it's a pretty long time. And what you may see is that in control mice, the levels are pretty small. However, in the challenged mice, you may see in red increase of IgM and following increase of IgG in the violet. And when we think about these patterns, they are very typical for many, many different antigens. So it's nothing very specific or strange or different to what we know in the responses in general. And we know that this type of response is T dependent, and usually it applies to proteinous agents like phages are. This is how it could look like if it was T independent. So how does it happen? It happens by cooperation of B cell and T helper cell. So we have this B cell that captures phage antigens, process it, demonstrate, and get support for the response from T cell. And in this situation, we can observe this typical pattern of increase in antibodies. And what is important here is that this type of response is linked to high affinity variance production, because this help from T cell allows B cell to maturate their affinity. And the other thing is that it promotes immunological memory. So we may expect that once we produce high levels of antibodies that recognize specific phage, we may expect that this response will be active for longer. And now a little bit of so what. We have observed it in a mouse model. Here you see that the mice were challenged with the phage and then used in the experiment in the time where we observed peak of IgM or IgG. Of course, here it's not perfectly isolated from IgG response, but still IgM prevails. So the challenge, and the challenge, and then investigation of phage circulation in these animals. And what you can see in this panel, everything, these are concentrations of phage in blood and in tissue. And everything in blue, these are control mice. So how much phage survived in control mice. But what we see in, let's say, pink, this is high IgM mice concentration of phage. What we see in red, this is high IgG group of animals. And as you see, particularly in blood, we have way more phage in control mice, less in IgM, and no phage survived in highly immunized mice with high levels of IgG antibodies. A little better in tissues. However, again, you may see significant differences, and usually we have way less phage in the immunized mice. And now about oral treatment where we observed the effect of second dosing, because a typical question is if we can give the phage again when we wait long enough for the decrease of antibodies. So we observed that in IgA, IgG does not tend to decrease in mice in the time of experiment. So, well, they don't live that long, I think. So what we tested, it was 100 days of treatment, then 120 days without phage, and then 44 days of phage treatment, which was the second dosing. Mice were treated orally, which is not very aggressive way of treatment. And of course, it was repeated, so this is how long it took. And this is what we got. So in the upper panel, you may see a IgA that was observed in feces. I'm sorry, I think that I must correct. So in the upper panel, this is how much phage survived in feces. And in the lower panel, you may see fecal IgA. And let's have a look at fecal IgA first. There are two different groups of mice challenged with two different phages, A3R in blue and 676Z in green, and of course some control mice. And as you see, we observed very slow increase, it's more than 3 months, of fecal IgA that was specific to the phage given in the drinking water. And more or less here, at day 40, we achieve high levels of this IgA. And when we go to the upper panel and we observed how phage survived that, we see that in the time when this was low level of IgA, we had high level of surviving phage. But when we had high levels of IgA, then we had only single animals with low concentrations of phage that were able to survive. And then 120 days without phage in the diet, it allowed us to get this decrease in fecal IgA. And again, the time where animals were exposed to the phage, at the very beginning, where the levels of IgA were still very low, we observed that these levels of phage that survived were more or less detectable. Yeah, so we got some phages that survived within more or less 2 weeks. But when these high levels of IgA came back, and it of course happens way faster when we have a second dose and already immunized animals, then we again see no phage that survived this gut transfer. And to give a summary, one thing is that non-specific immune response is the first mode of response that, however, strongly affects phage pharmacokinetics. Then specific immune response. It requires enough time and dose to develop, but its effects can be really devastating for the phage. And taking it together, phage pharmacokinetic is key to phage bioavailability, and therefore it determines the outcomes of antibacterial therapy with bacteriophages. I would like to acknowledge my team who worked on the experiments that I presented to you, and thank you.
Dr. Dąbrowska, thank you so much for a fascinating talk, getting at the root causes of resistance to phages. Thank you very much for all the work you've done on phage pharmacokinetics. So we'd like to open it up. Sure. So we'd like to open it up to questions now. And David, I don't know if you want to take the first question from the Q&A. We have many questions there, and maybe we can just take them for a few minutes if that's okay with Phage.
Yeah, I mean, there's a load of really interesting questions. I don't think we'll be able to get through them all. But there's I think first of all to Aycan. Does the Food and Drug Administration in America, I mean, they're often seen as key to getting any sort of treatment license on world markets, even outside the United States. Do they have any views, or have they made any statements about phage therapy? You mentioned earlier on from the 1930s, 1940s, articles from JAMA, but what's their current position in the US regulatory circles? Do you have any information on that?
In the US regulatory, I think they have been using commercial like phages for like in food industry, I think from like 2000s, the food industry settings. But I don't think they have been using like clinical or environmental like under the license. I mean, just the supported cases and clinical trials going on. So
yes,
yeah.
So you mentioned some kind of individual cases of phage therapy, and I know that almost kind of like Hail Mary ones, you know, the last throw of the dice, the last chance, people have used them. But
But I know that there are some like projects. I also off the record know that some clinicians also use phages, but of course they can't, you know, publish them. But only I think food industry, they use food industry as a disinfectants.
Yeah.
But there are some big projects, and also there are some grants. I believe that in America, I think that phage therapy is going to be more like common, but in the future, not now.
Great. So maybe I can take the next question. Maybe both of you can comment very briefly on this critical question. Will phage therapy become a primary treatment for bacterial infections comparable to antibiotics? Or do you see with what we know now that it will remain primarily a complementary treatment in addition to antibiotics? And I know that's a complicated, big question, but maybe you can both briefly just comment on that with what we know now.
Maybe I actually, in my opinion, it really depends on the infection. I mean, for example, in the case of like pan-drug or maybe multi-drug resistance bacteriophage therapy might actually become the primary treatment option, or in some specific cases like chronic or local infections, it might even, you know, become the primary option. But in general, antibiotics still have major advantages. They are easier to produce, store, and distribute, and they have strong commercial infrastructure behind them. I believe that, again, without centralized and accessible phage library, it is very hard to make phage therapy as first-line treatment. So now I think phages are powerful allies, but not a like universal replacement. This is my idea.
So I very much agree with what you said. The only thing that I would like to add is that it's very good to combine phages and antibiotics for one reason, because there are observations that this decreases the chance of bacteria to develop antibiotic resistance, because phages, they exert a specific biological pressure to their bacterial hosts, and these hosts may very often not be able to support mutations that are linked to antibiotic resistance. So of course, it's not, it's very individual. It depends on the phage, it depends on bacterium, but it's an option. So like double pressure is more than one, and it's very often very useful in a clinical approach. But again, I very much agree that we'll never know. It depends on the phage. I still hope that we will be able to buy at least some specific phages in pharmacy. I don't know how about engineered phages, which are an option, but there are a lot of criticism and doubts, which are, to my opinion, should be considered, that this is GMO and not really, and not necessarily they should be distributed without any regulation. So we will see in the future.
So, Aycan, can you say something about the current state of play of clinical trials in humans? It's not straightforward, is it?
Yes, actually, yeah, there are some clinical trials, but not a big numbers, just like 100%, I think. But just there was a publication, I think, published in Nature, 100, I think, includes 100 patients, but in like, I think, sepsis patients, and they use the phages IV, and the results very good. And also there is a big grant in Horizon program, and then they also, European Union support these clinical trials, but the collaborative one, not the for one country. So I can say that, like, you know, like this is a big clinical trial and the result is. Like small clinical trials getting there. For example, at the moment in Turkey, we are doing a clinical trial. It is going to be the first clinical trial in Turkey, human clinical trial.
Right.
So, and the results good, you know, so far. There are some separate cases, but it is going to be more and more, especially with the big like grants from the European Union and America and other countries. So, but at the moment, I can only say the one like big clinical trial. That's it.
Because when we're dealing with antibiotics, antimicrobials, these trials, relatively straightforward in principle, they still cost vast amounts of money. So, you know, I think it is a great challenge to set up a rigorous trial that will really satisfy everyone, including the hard-line skeptics. So it'd be very interesting to hear what's happening, what you get from the trial. What is the trial in Turkey? What are you looking at?
Urinary tract infection. Actually, we are using urinary tract infection, and then we are like apply phages through intravenous catheter, you know, urinary catheter. But I just would like to add something. I worked in TÜBİTAK, as you mentioned. There are lots of project application related to like animal trial. I don't believe that we are in that position. You know, phage therapy has been used in like over a million people in like former Soviet Union, but we have already huge experience. So there is no need to, you know, do like, let's do the animal trial again. I am not like agree with that. We just have to show that, as you said, rigorous and the good, well-designed clinical trials. Is it effective or not? It even is effective as antibiotic. That is enough, I think. We don't expect, okay, it is more powerful than antibiotics because we are trying to use, you know, make the antibiotic usage lesser. So I think we just need clinical trials to show, okay, safe, no side effect or minimum side effect, and also at least as effective as antibiotics. I think this is our primary concern.
Yes, absolutely.
So maybe my last quick question from me. I think, David, we should probably end in a minute or two. So maybe another question from you afterwards, if you think time allows. So quick question that I've been interested in for some time, and we have 2 or 3 questions on this from the audience. What about using phages in immunocompromised hosts? Often our antimicrobial resistant bacterial infections are in immunocompromised hosts, and I can imagine there's a risk of potentially different side effects in the immunocompromised receiving phages. They may have different reactions, number one, and number two, they may not be as effective in those patients. And so what do we know about the use of phages in immunocompromised patients? Dr. Dąbrowska, maybe, or both of you can answer.
Well, maybe Dąbrowska, please. Yeah.
Thank you. So I've talked a lot about the immune system that neutralizes the phage. So it's a very good and logical question. How about immunocompromised patients? Maybe it's better because they don't respond to the phage. Unfortunately, it's not. There is a very nice paper on that that comes from the Barbier group from Paris, and they observed that more or less in general, immune cells, they are necessary to help and to help in clearing the infection when we consider phage therapy. So they just demonstrated that. So I think that in an immunocompromised phage, on one hand, we don't have this this induction of phage-directed responses, but we also don't get this help that comes from the immune system, and which is known to be very important even in antibiotics. Yeah, like we, for instance, use bacteriostatic preparations, which do not effectively kill bacteria, but we still can clear the infection. And it's a very good example how important the immune response is in that.
Okay, so there may be some difficulties using it in severely immunocompromised hosts, like perhaps transplant patients. But it sounds like we may need to have separate trials or separate approaches for those patients. So that's a very interesting aspect to the introduction of phage therapy in the world. Thank you for your response. David, do you have maybe a final?
There are plenty of questions to ask, not also on the questions that have been sent in, but also questions that I'd like to ask. But I would get into big trouble if I carry on, I think. So I think at this point we do need to draw the webinar to a close. It's my great pleasure to thank both speakers, Dr. Aycan and Krystyna. I think you've given some really great presentations today, really clear, very thought-provoking, and a wonderful webinar. I'd like to thank Michael, my co-chair, for supporting here, and Phage for doing all the behind-the-scenes technical work. And just to say to everyone, if you look at the ISAC World website, we do, after a few weeks, have the webinars put onto the ISAC website. They're available to view again, or if you haven't seen them at all, you can view them for the first time. And they're there for a long time, and they're always free to view. So please visit the website and have a look at what we've got on there. And at that point, I say thank you to everyone, and hopefully see you for the next session, which again you can see on the website when that is. Thank you very much indeed. Bye-bye.
Thank you.
Thank you.
Thank you.
Goodbye.
Bye.